So I welcome everyone to this session. My name is Valérie Schnicker. I'm a professor of pharmacology of Strasbourg University, France, and I will share this session. It is a great pleasure and honor to introduce Professor Huang. He's a professor of biomedical sciences and vascular biology, and also Janie Hu, professor of biomedical sciences, and the head of the Department of Biomedical Sciences at City University of Hong Kong, China. Professor Huang obtained his training at Shanghai Medical College of Fudan University and received a PhD from the University of Cambridge in pharmacology. After that, he validated 2 postdoctoral trainings, one at the University of Vermont and one at the University of California in LA. And his major research interest is focusing on vascular biology, on physiological and pathological conditions. He has a major interest in endothelial dysfunction, early plague and development of cardiovascular disease, and also in the development of the different steps leading to an atherothrombotic events, and also vascular inflammation, which is closely linked. The topic today, his presentation, is entitled Haemodynamic Regulation, Energy Metabolism, and Vascular Homeostasis. Please, you.
Thank you very much, Valerie. Good afternoon, ladies and gentlemen. I'd like to thank David and also Amrita for the kind invitation and also organizing this wonderful the 19th World Congress of IO-Pharm. Some IO-Pharm are from Hong Kong and China. Today, I'd like to share with you some of our recent work on the impact of hemodynamic forces on vascular function. Now, it has been known for many years, and there's a distribution of atherosclerotic plaques along the entire arterial trees. Actually is not evenly distributed. And you see this area, circled area, actually is, you find more formation of those plaques. And this is mainly due to this different pattern of the blood flow we call disturbed flow, which actually is pro-inflammatory. I will explain to you a little bit later. And then more straight part, you find very few plaques actually can be localized in the area. And those are the area we call the endothelial cells in that area of blood vessels exposed to the laminar flow. So that means this flow will produce a protective effect on endothelial cell function. So here you can see very clearly now in this area you see these little spots. These are actually deposits of plaques, and they mainly form now in the inner curvature or this bifurcation of the aortic arc as compared to the high stretch area outer the area of aortic arc. Now, this can be explained partly by the very unique impact of 2 different patterns of blood flow on the endothelial cell property or phenotype. Now, just briefly introduce it to you. Now, in this disturbed or turbulent area, endothelial cell actually poorly organized, and then you look at this orientation is not in the direction of blood flow. And instead, and this the cell contact among this endothelial cell loosen up, increase permeability. And then if you measure the level of oxidative stress or the expression of pro-inflammatory genes, they all are the increase. Now, by contrast, endothelial cell in this area, straight arteries, actually, they are well organized and they align in the direction of blood flow. Now, they have very high permeability, very low permeability, and a very low level of the oxidative stress, and also the lower level of expression of those pro-inflammatory genes. So that's why now in those areas, very few atherosclerotic plaques are formed. And this is probably this basic fundamental this explanation now for this uneven distribution of plaques now along now these two different anatomical locations. So just to give you an illustration here by one of my former students. Now, this aortic arch, in general, they're exposed, more often exposed to the disturbed flow. And then we able to imagine this, the expression of VCAM, the vascular cell adhesion molecule. Actually, it's an important molecule. Initially, they attract these circulating monocytes during the initiation of atherosclerosis. Going too fast. All right, but now just you get a little bit idea about, the plaques normally is form in this the branching points of blood vessels, and then we know quite well now disturbed flow leading to now increase the endothelial cell inflammation and also increase production of reactive oxygen species, and that closely linked to the atherosclerosis. But the detail these molecular events now along this the process are not really clear. But we know there's any pattern of shear stress or different type of blood flow I mentioned here must be sensed by the proteins expressed by the cell surface on the lumen side of blood vessel. And we were invited last year to write a review article now about these various proteins expressed by endothelial cells. It doesn't matter receptors or ion channels, normally non-selective cation channels, and also some enzyme. And all these are able to pick up some flow signal or shear force, and then transmit it in the end, acting on now this a variety of mechanosensitive transcriptional factors. And I think this is a very complex picture now about this, the mechanosensor on the endothelial cells and the leading to change of gene expression related to inflammation and atherosclerosis. So today I would like to give you two examples, two or three. One is called YEP and the TEST, which actually is the effector of Hippo pathway, I will mention later on. Another is the KF2. It's a very important transcriptional factor in endothelial cell, controlling activity or expression of nitric oxide synthesis in the endothelial cells. Now, what is a Hippo pathway? Now, Hippo pathway actually is highly conserved across different species, and which consists of cascade of kinases. And the last effector actually is a YEP or TEST, and normally contained in the cytosol. But this can be altered in response to certain, there's stimulations. And for example, now, this is highly mechanosensitive, and a lot studied in cancer biology because they control. Now, this pathway important in controlling the organ size by balancing cell proliferation and also apoptosis. But no one actually knew until we start to look at this, whether or not endothelial cell express this hemo pathway able to respond to the mechanostimulation. Now, how you determine the activity of YEP? Just very briefly. Now, if you switch off hemo pathway, what will happen? And the phosphorylation of YEP in cytosol will decrease. And the decrease, that means will promote the nuclear translocation of YEP. And the YEP at the cell is not transcriptional factor, but it's a co-activator of transcriptional factor called TET. And then once this interaction leading to increased TET activity and increased expression of those well-characterized these target gene, and these two I will mention to you. Now, if you turn on this Hippo pathway, then what will happen? Increase phosphorylation of YEP. Now, this is increased PYEP will be degraded. So that means you have a less or reduced amount of cytosolic YEP, and then you will reduce nuclear translocation. Basically, TET not going to be activated. So the target gene expression will be suppressed. And this is bad. That means promoting cell proliferation and increasing inflammation. And this is good. If you turn on Hippo pathway, and then will reduce expression of those target genes, and then which actually we found important in inducing inflammation and atherogenesis. Now, at that time, no one really knew whether or not these two types of the shear stresses or this flow pattern have any impact on the endothelial cell Hippo pathway activity. And then we have no any idea whether or not this Hippo pathway can be affected or by any of those factors which actually associate with the atherogenesis. And then we try to address this question, and I just show you some of the data. And this is Dr. Huang Li. At the time, was my PhD student, then postdoc, and actually spent about 2.5 years trying to understand now how this YAP activation involved in the generation of the plaque. And then what we find, as I mentioned, this is mechanosensitive. So we use our tools. For example, as I mentioned, we dissect piece of this aortic arch, which normally exposed to disturbed flow. Is not good. It's pro-inflammatory. And then thoracic artery is more straight, and then which actually exposed to laminar flow, which is vasoprotective. So you can see here clearly. Now this AA, aortic arch, so you have less the P YAP, the phosphorylation. That means the YAP activity will be increased, yeah? And as compared to thoracic, this aorta. And this is highly repeatable. Now, if you, and this can be further illustrated by the end-phase staining of YAP, and you can see now this inner part of aortic arch, you have a more nuclear translocation of YAP. So that means the YAP now start to turn on TET and increase expression of those inflammatory genes. So initially, of course, we identify this YAP mainly confined or localized in endothelial cells here. You just dissect a piece of aorta from normal mice. And then, now this is a contained endothelial cell. You see this increase, this high level of expression of YAP, and the enos as a marker for endothelial cells. Now, in another piece of artery with endothelial cell removed mechanically, and you see this is a markedly reduced expression of YAP, and together with loss of enos. So in order to dig up this molecular mechanism, we need to use now this need to perform a lot of this in vivo the cell studies. And what we use actually this is called BD, cell culture flow system. We can grow cells here, and then we can mimic this either disturbed flow or the laminar flow. So here you can see very clearly, if you subject endothelial cell to the laminar flow, and then what will happen? And within 15 minutes, you can see a rapid increase in the YAP phosphorylation. And this increased phosphorylation can maintain until 4 hours. Yeah? And that's for quite a long time. Now, by contrast, if you expose cells to disturbed flow, then what happens? And then rapidly, this is phosphorylation of YAP, and then rapidly fall, and then about, the gradually coming back probably after 1 hour. And again, now this is target gene of YAP and can be differentially regulated. So you have a laminar flow, you can suppress expression of these target genes, but the disturbed flow able to increase expression of these target genes. And also in the end phase, and the cells very clearly, you can see this YAP actually, in response to laminar flow, and YAP mainly contained in the cytosol of endothelial cells. Now, there's another way to demonstrate the clear impact of hemodynamic forces. Actually, is shown in this experiment. This is in vivo. This is a mouse. This is a rat abdominal aorta. And then we did the surgery, And we try to narrow a little bit, use a cleft. Now, somewhere along the abdominal aorta. So the flow in this direction, and the normally flow in this direction, this is laminar flow. But when you have a narrow this area of blood vessel and increase the speed of flow, so that means increase shear stress, and then possibility of producing greater beneficial effect. Now downstream here, probably this low shear stress or might be turbulent flow. So we're able to stain. Now this is a phosphorylation of YEP on the endothelial cells. You can see cleft area, you have an intense staining of this YEP, the phosphorylation. So that means have a suppressed YEP activity. And then upstream, you have a little bit more staining, but downstream endothelial cell has a very weak staining of a PEP. But of course, we need to dig out what are those targets involved when you turn on or activate YEP and test the pathways. And then we constitutively activate the human endothelial cells and subject to the RNA sequencing. And then we analyze this pathway and enrich. You can see here, there's a couple pathways are closely related to atherogenesis. Now, one is about this leukocyte migration. Another is cell-to-cell adhesion. So these can be confirmed easily. Now, in the monocyte attachment, the experiment, if you overexpress the YEP in the human endothelial cells and then perform this attachment experiment, you can see very clearly now, after this activation of YEP in endothelial cell, now this human monocyte attachment to the endothelial cell rapidly increased. And that is very clear to us, and the YEP actually is very important in initiating atherosclerosis. So this can be confirmed in ApoE, these knockout mice, which actually is prone to the development of atherosclerosis. And then we specifically activate endothelial, this YEP, and then put them on the the Western diet or high cholesterol diet for a certain period of time. And you can see very clearly here, it's oil staining. They indicate amount of the plaques formed along the vascular tree. So you can see very clearly this EC-specific activation of YEP and the increase now the formation of atherosclerotic plaques in these ApoE knockout mice. On another way, we also able to generate an animal model. Actually, those are related to disturbed flow and enhanced atherosclerosis. And then what we did, actually, you can partially ligate this left the carotid artery, and then put this animal, this is ApoE knockout mice, on the Western diet for 3 weeks, for a very short period of time. And then you can see these white spots actually is a buildup of plaques. Yeah? And this area actually is disturbed because blood flow now, because this narrowed the area and then flow become disturbed and just in this area. Now, we use a CRISPR Cas9 genome editing technique and selectively knock down the expression of endothelial YEP, then what will happen? Now, use this model, and you can see this verified by reduced expression of YEP in the aorta, and also the plaque formation actually markedly suppressed. And this is another piece of useful information to demonstrate. Now, this hemodynamic forces now has a very important impact now on the formation of plaques, mainly through regulating the YEP activity in endothelial cells. Actually, Huang Li spent about over two years this intensively, because we try to figure out this detailed molecular mechanism inside the cell once the endothelial cell sense laminar flow. And I very briefly explain to you now. Actually, we identified this integrin. Actually, integrin is a mechanosensor, but a different isoform. Now, integrin beta-3 able to sense now laminar flow. Once this activates, recruit this small g protein called G alpha 13. And this form a complex suppress low A. Low A already been known long time ago. Those are pro-inflammatory. So low A is able to control this YEP activity because they control YEP phosphorylation. If it's too much low A, actually this YEP will be dephosphorylated. And the more YEP will enter the cell, enter the nucleus, turn on the expression of pro-inflammatory genes. So I'm not going to go detail, but we are very pleased. Actually, our report was commented in Nature, News, and the Views. And also I like this title called Turbulent Path to Plaque Formation. Now, one of our research objectives actually try to identify new therapeutic targets for drug development. And initially, or naturally, actually, we screen a number of those lipid-lowering drugs, and then we show only the statins. There's several different types of statin. They're structurally different, and they're very potent to suppress now this YEP activity. Not those other, for example, niacin also lower the lipid, but it's very minimal impact. Now, this study help us to build up this YEP-targeted drug screening platform. We have screened this over thousands FDA-approved drug library and identify quite a number of those drugs with a potent inhibitory effect on the YEP activity. Just give you one example. Now, this is Curexin, CBL. Actually, it's anti-tumor drugs, mainly for the treatment of a solid tumor. And these drugs is very potent to suppress the YEP activity. Now, we have both in vivo and in vitro experiment, very briefly. Now, if you expose human endothelial cells now to these drugs at a low concentration, I think as low as 1 micromolar, and you will see this is another type of phosphorylation, another the amino acids, the tyrosine, and then you see there's a rapid decrease about 30 minutes after drug exposure. Now, without affecting another phosphorylation site, which actually we reported in our Nature paper. And very clearly, these drugs can suppress expression of all these YEP and the target genes. And it doesn't matter in the basal level or in the cells exposed to those interleukin-1 beta, for example, shown here. Now, in the animal study, we can confirm this. So it doesn't matter in the female or the male or female ApoE mice. And then we can show very clearly, now this is 1 milligram per day, the treatment for quite a long time in this particular study. And then, again, we use oil red staining to measure the formation of plaques, and then in both strains, the sexes. And actually, these drugs is quite effective to reduce plaque formation. Yeah, but I do not have time to go through these the mechanistic details. But this is one of example. Actually, we have a couple other drugs has a different mechanism, but they have a very potent the YEP inhibitory effect. And this is just recently published by my final year PhD student in BJP. Now, I'll move on to because talking about this mechanosensitive regulator. There's another one actually is very new to us. It's actually mitochondrial uncoupling protein 2. Because UCP2 is very important in controlling mitochondrial oxidative stress. And also in the early on, actually about 10 years ago, we published the first paper probably about UCP2 important in keeping, restoring normal endothelial function in obese, diet-induced obese mice in circulation research. But no one really knows whether or not this, that the shear force can have any impact on the function and the expression of UCP2. So we have just recently published a paper, and I also like this title called endothelial UCB2. It's a mechanosensitive suppressor of atherosclerosis. So here, I think this is very useful to use it's a BD flow system, and then we can detect there's any proteins, any targets you want. Now here again, use human endothelial cells. If you expose your cells to the laminar flow or unidirectional shear stress, and you can see there's a very marked increase in the UCB2 in endothelial cells. And also, if you expose your cells to the disturbed flow, and then this expression of UCB2 actually is suppressed. So this is very clear. Yeah, that's very clear. And this is also the first piece of information. Actually, UCB2 expression can be regulated by the mechanismal forces. And then we again use this very unique tools, and then we're able to dissect very small amount of tissue in the aorta. And in this case, for example, the aortic arch and also thoracic, the aorta, and you're also able to see a very different expression of UCB2. Of course, now in the straight part, you have a higher expression level, and compared to the UCB2 content in the aortic arch, where it's more disturbed flow region. So we try very hard because UCB2 is actually the proton channel or transporter in the mitochondria. It's not transcriptional factor. So we try pretty hard, and then what are the transcriptional factor which happen to be mechanosensitive and regulate UCB2 expression. And we screen quite a number and identify this KLF2 actually is upstream regulator of UCB2, directly interacting with some the promoter sites of UCB2. So here you can see very clearly now. Now this is the flow here, and then without this activation of KLF2, and then you will see very clearly UCB2 will be suppressed. But if you overexpress KLF2, and then this will reverse this effect of disturbed flow on the UCB2. Or another way around, if you can, the laminar flow increase UCB2, which can be reversed by the suppression or the knockdown of the KF2. And this is a very clear link here. It doesn't matter how you measure the messenger RNA or proteins, and always same. So here we establish laminar flow actually through this KF2 and increase UCB2. By contrast, disturbed flow actually able to suppress KF2 and then reduce expression of UCB2. Now, of course, again, because we are more interested in there's any drugs or chemicals or small molecules actually able to act on this signaling pathway. And then we happen to identify a few, and then one is starting again. Starting is very, very interesting and important molecules, which has so many different beneficial effects. Now again here, you can see very clearly now, if we use interleukin-1 beta, which suppress expression of UCB2, and which can be rescued, for example, now by these the statins or resveratrol, and which is another compound has multiple action sites and multiple beneficial effects in many different animal studies. And I do not need to go through here. And then actually we have this few, and we actually also test a number of other compounds, and some happen to be also able to increase the KF2 activity. And this is well confirmed now, and then this vasoprotective drug, and able to increase UCB2 mainly through increased activity or expression of KLF2. That this again, we always try to validate in the cell study, cell results in the animals in vivo, and then we can rapidly generate the plaque formation by overexpressing the PCSK9, which can degrade liver, the LDL receptor, so that more lipids will stay in the blood, and then that will contribute to the rapid development of plaques. And here you can see very clearly now, if you suppress or the inhibit the endothelial cell, now UCP2, and actually you will increase the plaque formation. Yeah. So we're able to link so this CAV2 and the UCP2 in the animal level as well. So very brief summary for this part of work. So we have a flow, disturbed flow, the laminar flow again, and also starting resveratrol, all able to increase expression of CAV2, very important. This the transcriptional factor in endothelial cells. And then these have a direct link to UCP2, and actually it's a certain area in the promoter, and actually there's a direct interaction, and the increase UCP2, the expression. Now once you have a UCP2, the expression increase, actually there's another, the bad guys can be inhibited. We call factor 1, and the factor 1 itself actually turn on the inflammation and accelerate atherogenesis. But if you UCP2 increase, and probably through MPK, another important vasoprotective regulator, and then suppress this, and then reverse inflammation and oxidative stress, and slow down the development of atherosclerosis. Another interesting story probably you like to listen is the role of CAV2 in the vascular calcification, because no one actually look at this the impact of hemodynamic forces on the vascular calcification. This is a quite interesting study I share with you. We know that vascular calcification is a very important topic in this cardiovascular research, because there's many diseases that vascular wall actually calcified. Yeah? But no one really know whether or not this is either disturbed flow or laminar flow able to control or this regulate the process of vascular calcification. Now again, we find this CAV2 mainly expressed by endothelial cells. You can see here, this is aorta. If you remove this endothelial cell, the expression level of CAV2 is very, very low. So that makes sense, because this only those the transcriptional factor in the endothelial cell, because able to respond to the shear force, and then mainly should be mainly expressed by the endothelial cells. And also in this cell culture system, we're able to demonstrate now CAV2 can be rapidly increased in cells in response to laminar shear stress. And this can sustain for quite a long time, but in our case, 24 hours. But I believe they can sustain probably longer beyond these 24 hours. Now, what's a CAV2? CAV2 is actually well-defined, characterized before, as a mechaноsensitive transcriptional factor. And most important thing, of course, if you get increased the CAV2 expression and the increased eNOS activity, increase production of nitric oxide. So that means locally will increase vasodilatation. Doesn't matter what animal models you use. And this is known, but only thing is unknown, actually, we want to study is the role of endothelium. CAV2 in vascular calcification is unclear. Now we happen to get this human, the human aortic valve. The valve when they were open, and then ventricular ejection, they will eject certain amount of blood into aorta, and those are unidirectional. There's a flow, and those you can see on that side, ventricular side, there's very little calcification, because endothelial cell in that area always exposed to laminar flow. But when the valve closes, aortic valve closes, and a certain amount of blood will flow back, and they will hitting the wall, and then you can see now aortic side of this valve now contain a lot, these calcified, the spots here. And this is highly repeatable in almost all the aortic valve we collected from hospital. Now, today we're talking about the role of CAV2, and that we're able to use those samples. Now, this is, for example, this is a control, this is calcified, the aortic valve. This is endothelial cell, probably partially turned into other cell type. I do not have time to explain to you. But when you stain this presence of CAV2, you can see very clearly. Now, in the calcified, now this aortic valve, now this is the intensity of CAV2 much, much reduced. So that's very clear to me. There's a link between now the expression level of CAV2 and also proper endothelial cell derived calcification. But this can be partially confirmed by our cell culture system. But here we build up this special chamber, and then we grow endothelial cell, and then allow this the culture medium flow in one direction. So 1, for example, 4 and the 4 and the 6, certainly this laminar flow just mimic the straight portion of our blood vessels. And then this branching point, this curved, and merge into the one. And all this area that's supposed to be affected by distal flow. Now, if we use this also sensing, this dye, and then able to detect the imaging, this calcification. So you can see very clearly in this area 2, 3, and 5. And then you see this little red spots actually is calcified, area. And this can be confirmed in our human endothelial cells. And then more interesting, actually, we're able to collaborate with our the radiologist in one of the hospitals in Nanjing in China. And then he imaging this human coronary artery. And then we pick up these 4 important coronary artery, and then we'll measure these calcium scores, the calcification scores, and then we'll estimate. So you can see this bifurcation area, you always have a higher calcification score as compared to the bifurcation area. Yeah, this is from 48 patients, I think. And this is very clear, we have a summarized data here. So to me, of course, we do not know, but the calcification now in the human, those arteries is due to endothelial cells or median muscle cells, we cannot tell. But at the least, because in this area, they have a different pattern of hemodynamic forces. So again, we use ApoE knockout mice on the Western diet for a certain period of time. And then not only this, branching points or aortic arc grow more the plaques, and then also more calcification. And then we're able to use osteosensing to show this. And then all different areas we characterize and then define. Now this is area with distal flow now have a high chance of developing calcification. Now we know there's a term called endothelium to mesenchymal transition, actually one of proposed important mechanism now in the development of vascular calcification. But actually, if we expose yourselves to the distal flow, you can start to see the expression or elevated expression of markers for this mesenchymal, the transition. Now, if you silence or suppress expression of CAV2, you can repeat almost identical those results in response to distal flow. Just very briefly here. Now, if you suppress expression of CAV2 and trigger this MT, and then all these markers increase, and then turn into the media mesenchymal cells, and then these mesenchymal markers increase, and then further, of course, osteogenic differentiation. Now, all these, the osteogenic marker expression increases. But in the end, you're able to stain now this calcium deposit in the culture endothelial cells. So this is probably very likely this process involved once you have reduced or the suppressed expression of CAV2 in endothelial cells, and then leading to increase the calcification in the blood vessels. Now, validation, again, this in vivo validation for the role of CAV2, and very briefly. So if your endothelial cell specific knockdown of CAV2 and able to increase vascular calcification. And all these markers actually can be confirmed and very clearly in this kind of experiment. And by contrast, if you use AAV-mediated endothelial CAV2 overexpression, then you're actually able to reduce now this calcification and also suppress now the expression of osteogenic markers. Yeah. Now, of course, you would like to ask about what are the underlying mechanisms. Actually, we overexpress the CAV2 in the endothelial cell, and the same for this RNA sequencing. And this is the further analysis. We identify actually this CAV2 overexpression able to suppress BMP and the SMAT signaling pathway, which actually is known before, and either in endothelial cells or muscle cells involved in the development of calcification. So I do not need to go detailed, as we spend quite a lot of time to study this, particularly now in the cell studies. And this actually is one of the important mechanisms also shown in the title of the paper. So this is a small summary. I have a couple more slides to go and then finish. And this red line actually is novel findings from our study. And then this actually is quite interesting. And I would like to thank these few members, some are students, some are postdocs, for involving this study. And again, we really want to identify any drugs, any drugs able to increase more specifically the endothelial cell CAV2, whether or not that can be potentially used to slow down the development of now vascular calcification. Now, a couple more slides before I start. Now, because we today talking about the hemodynamic impact on the vascular function, and the one way naturally, actually, when you're doing exercise, increase activity, increase this heart rate, and then it will increase blood flow, increase shear stress. And then many years ago, actually, not many years ago, 2017, and then we published a first paper probably quite detailed in both conduit and the resistant artery in response to physical activity. We put on this diabetic mice or induced obese mice on the treadmill, and then this exercise protocol is very moderate for about 6 weeks. And then what we're able to find now here, now for these obese mice, you have a very poor this flow medial dilatation, which actually in human be used to measure the function of endothelial cells. But in this small resistant artery, which is normally control of blood flow, and the resistance is very important in determining diastolic blood pressure. But here you can see now, this is a control, the DBDB mice, these DBDB mice here, and a very little this flow medial dilatation. So once you generate this pressure gradients between two ends, and this actually is a diameter of this very small resistant artery. This is mesenteric resistant artery. Now, after 6 weeks, this running exercise here, and you can see this marked recovery of this flow medial dilatation. This is very interesting because after I see these results, and I start doing a little bit exercise, but not that much, but I walk. And walk actually also help. They will increase your heart rate a little bit, and then increase shear force. But we continue on this study. But more recently, we just get paper accepted by diabetes. Actually, it is a CAV2 as well. And we find when you this increase exercise or physical activity, and then we're able to detect increased expression of CAV2 in endothelial cells. And there is very clearly now we're doing a lot of work and try to find out now how this KF2 in the end actually can increase now expression of nitric oxide synthesis in response to physical activity and then improve endothelial function, either in the diabetes mice or dye-induced obese mice. My last slide. Now, endothelial dysfunction actually is a very critical initial pathological event, now leading to a lot of different diseases. Of course, today I'm talking about the impact of hemodynamic forces on the endothelial function. But here again, now, this if endothelial cell cannot work very well initially or in the long run, leading to hardening of blood vessels or the development of atherosclerosis. So how can you overcome this? Physical exercise, I think, is important because now more and more Chinese like to do exercise. And of course, because you need to have time. But in Hong Kong, everyone works very hard and not much time and also limited space. But people are always able to find some time to increase physical activity in the weekends, for example, whenever they are free. So when doing exercise, your this heart pumping activity increases and increase stress on the endothelial cell, probably in the entire vasculature. And this is very important, as I mentioned, doing a lot of things. Yeah, increase KF2 expression, increase UCB2, which and suppress these the endoplasmic reticulum stress, which are bad guys in obesity and diabetes. So I think I hope this taking home message, everyone doing a little bit exercise, and that will effectively reduce the chance of developing atherosclerotic vascular diseases. So this is my slides, last slides. So I'd like to thank my team. This picture taken quite a number of years ago, and also collaborators mainly in Hong Kong and also in mainland China, a couple in other countries. And thank you so much for your attention.
Thank you so much for this beautiful lecture and letting us share your beautiful data on KLF and UCP2. There was a question, please.
Thanks very much, Yu, for your insightful talk. I have two signaling questions. First of all, your first part of a study, YAP, dephosphorylate YAP into the nuclear, associate with junk to regulate prone inflammatory downstream. So what is the downstream of junk to exert this function? Is a C-jun or C-cause? What kind of a transcript factor downstream of a junk?
Thank you much for the question. At JNK, we were able to spot the JNK. JNK was known for a long time to be pro-inflammatory, but we did not go the detail which, for example, those types, for example, JNK, because we stopped there. And I cannot remember there's anyone actually continue our work and try to find out more information about very, very downstream, yes.
Okay.
We did not do this.
Second question is, what is immediately upstream kinase regulate KL2 functions? Is a KL4 subject to phosphorylation or other kind of translation for modification move into nuclear for the function?
We did not really study that part here. We only use those known knowledge. And then we detect the changes in the phosphorylation of YAP in a couple of those amino acids. And then we use it as an index. Yeah. But we did not study those, how this phosphorylation occurred or those things, whether or not degradation, for example, retained by the cytosol. Those are a lot of studies in the cancer research area.
All right. Thanks very much, Yu. Bye-bye.
Thank you.
Yes.
So first of all, thank you for informative and fascinating talk. So my name is Xiaohua and I come from Mainz University. So in your presentation, you talk about one platform to screen out the YAP target drug. I'm wondering how you develop this platform. And my following question is that do you still have some following research about this selected compound?
Yeah, but these are very common, probably like any other lab with this luciferase reporter gene assay. And then now, of course, we have this platform extended to many other new targets we have identified. And the only thing is we need to identify this larger library, the drugs. But now we try to even able to apply probably to the, there's a metabolized in the human blood and extended. So now even in the urine as well, but we'll see. But we have identified quite a number of drugs. Some are very interesting, but I cannot tell you because we haven't released the whole entire study. Some are very interesting. Actually, some drugs, actually centrally acting drugs. Some receptors mainly expressed in the central neurons and also in endothelial cells, and that can serve as another totally new mechanism sensor as well. But we still continue on this, and then a lot of animal studies. Hopefully, within 1 year, we're able to release the results.
Okay, I'm wondering for the YAP target drug, is it a FDA approved drug or a new?
Yes, FDA approved. This is another question about repurposing those drugs. But of course, we now seeking for other drug libraries, small molecules. But this whether or not they can be developed further into drugs, but that will take longer years. Yeah. Repurposing probably easier.
Okay, thank you.
Thank you.
Go ahead.
Thanks. Great studies. With both of your targets, you've shown impressive effects on reducing plaque burden. But have you had any? Have you done any assessment on whether you've got any changes in plaque stability?
You mean this? We have this. We have some study on this, but I cannot remember the details. Yes, yeah. But currently we study more because we're actually interested in the secretory protein under control of this HIPPO pathway and where they produce, where they go. So we can now able to specifically label, this secretory protein from tissues, and that's you mentioned we can apply all those techniques to see the interaction of secretory protein or to the other membrane receptors or any other the interacting partners. Yeah.
Yeah, because if you could show, for example, that KLF2 is involved in improving plaque stability, that would be pretty exciting, especially.
This we have this data actually.
Yeah.
You can read our papers. We have this chip. We have this data. You can see very clearly because we're doing a lot of these deletions, mutations, and then we actually in the end narrow down certain amino the length of a nuclear basis, and then we know where they are roughly. Yeah.
Thank you.
Thank you very much. Time is already over, so we have to close the session. So thank you very much, Professor Yu Huang, and also the audience for attending and being so actively involved in the discussion.
Thank you so much. I hope I did not overrun the time. Thank you.
Thank you.